Welcome to Ethel Browne Harvey Postdoctoral Seminar Series. My name is Ekasit Sonpho, and I am postdoc at Stowers Institute of Medical Research, and will be moderating today with Katie Goodwin, the postdoc at MRC Laboratory of Molecular Biology. We are excited to highlight the work of our outstanding postdoctoral members. Today, Ankita Thawani from Baylor College of Medicine and Konstantinos Kalyviotis from Imperial College London will share their research. Each speaker will give a 20-minute talk followed by 10 minutes of Q&A. Please enter your question in Zoom QA box. For the first speaker, Ankita is currently the postdoctoral associate in neuroscience at Baylor College of Medicine under the lab of Dr. Andy Grove. During her graduate study, she received her PhD in biological science from Purdue University under the supervision of Dr. Donna Figetis. She received Bachelor of Technology and Master of Technology in Biochemical Engineering and Biotechnology from Indian Institute of Technology, Delhi, with minor degree in Management Study under the supervision of her master degree advisor, Dr. Saroj Mishra. Ankita received many awards and honors. During her graduate study, she received H.E. Embarker Outstanding Graduate Student in Research Award from Purdue University. In 2021, she received the Outstanding Scholar in Neuroscience Award from NIH. In 2022, she received both Best Doctor's Postdoc Award and Travel Award from Santa Cruz Developmental Biology. And today, she will going to present in the topic of the FOXI3 transcription factor is necessary for the phase restriction of placoidal lineages at the neuron plate border. Ankita, the stage is yours, and we are looking forward to your presentation.
Thank you so much, Ekasit, for that lovely introduction. And without further ado, I will go ahead into my presentation. Thank you so much to SDB Ethel Browne Harvey Seminar Series for this opportunity to share my work with you all. I study vertebrate neural plate motor development and how cell specification occurs in this context. During the evolution of chordates, the vertebrates form this complex new head. They develop complex sensory organs and eating apparatuses or jaws in the anterior parts of their bodies. And this enables the taxa to transition from a passive to an active mode of predation, and they become better survivors. And that is compared to largely filter feeding invertebrate chordates. When we follow those sensory organs and eating apparatuses, we find that the craniofacial region, the upper and lower jaw originate from neural crest cells, and the olfactory pit, inner ear, and the eye, specifically the lens, come from the sensory placodes or the preplacoidal domain. To give you a comprehensive list of cranial derivatives from crest and placoidal lineage, sensory placoidal domains invaginate or delaminate to form lens, olfactory pit, part of the pituitary ganglion, the inner ear, and several cranial ganglia. And the neural crest cells, they develop in parallel, they migrate in parallel to make much of the craniofacial skeleton in the head region. They add glia to the placoidal-derived sensory ganglia, and they also make a couple of ganglia themselves. When we fate map these two parallelly existing cell population, we find that they come from a thin region of the ectoderm, only a few cells wide around the stages of gastrulation, called the neural plate border. It's an overlapping region between the definitive neural plate and the non-neural ectoderm. And the small few cell thick region of ectoderm has the potential to make all the sensory organs, all of the craniofacial skeleton, and much more. In addition to the neural crest and preplacoidal progenitors, it is also possible that these progenitors retain the potential to make neural plate cells, as well as the epidermal lineages, meaning all four ectodermal fates in this tiny area. So what is the nature of the cell population? Fate mapping studies show So we think that we want to question where the placodal and neural crest cells arise and if they come from a common progenitors. And one hypothesis is that are the cells of the neural plate border multipotent, meaning do they retain transcriptional and epigenetic signatures of a mixed population with the potential to make any of the 4 ectodermal derivatives? Or is it the differentiation landscape is gradual with binary decisions of differentiation and bipotent progenitors? Our lab has reported a gene expressed at the neural plate border, FOXI3. In the mid-gastrulation embryo, around E7.5 in mouse development, we find that FOXI3 is expressed adjacent to the neural plate. The neural plate here is labeled in with SOX2 in red, and flanking that is the GFP expressing region representing FOXI3 expression. The left video is a whole mount, and the right video shows anterior to posterior optical sections throughout the embryo. And you can clearly see the green expression adjacent to the red SOX2 expression. Because the neural plate border is such a dynamic state, we sought FOXI3 expression at the neural plate border using this novel in-house made FOXI3 GFP mouse line, and we studied that spatiotemporally along the gastrulation and neurulation ages. We find that the expression begins around embryonic day 7.5, and it diminishes significantly by embryonic day 8.5. And at the middle of this window of expression, we chose E7.5 for subsequent experiments, which is the peak of FOXI3 expression. And it is indeed a key stage when the neural plate border or the neural folds begin to become apparent. At higher magnification, we observe strong FOXI3 expression adjacent to the SOX2 positive neural domain or neural ectoderm, and we find overlap of green and magenta double positive cells as shown in this magnified image. We co-stain these embryonic sections with AP2A, which is a known non-neural ectodermal marker, and this also shows an overlap with SOX2 and extensive co-expression with FOXI3, and this is the neural plate border. So FOXI3 is expressed at the right place at the right time. So we ask, could FOXI3 be used to identify, isolate, and query the cells at the neural plate border? To explore the role of FOXI3 in ectodermal patterning, we made another novel mouse line, a conditional Cre recombinase construct cloned into the FOXI3 coding region. We mated these FOXI3 mice to ROSO26 TD tomato reporter mice we obtained from JAX and delivered tamoxifen at embryonic day 7.5 when FOXI3 clearly labels the neural plate border to activate Cre recombination and thus activate the TD tomato lineage tracer. And we follow FOXI3 positive neural plate border cell lineage. We analyzed serial sections through the embryonic head at embryonic day 7.5 and found that robust TD tomato labeling is observed at the sensory placodes. An example of lens, olfactory placode, adenohypophysis, inner ear, and several cranial ganglia are observed here. And you can see strong TD tomato labeling in the placodal derivatives only, and some TD tomato labeling in the epidermis, as shown by these arrowheads, but no TD tomato red signal in the mesenchymal tissue around the placodal derivatives. So we wonder, maybe at this age, we missed an earlier window of FOXI3 expression where neural plate may have contributed to all 4 ectodermal lineages. And to test that question, we activated Cre-ER at stages earlier and later than embryonic day 7.5. And we find that embryonic day 5.5, Cre-ER activation shows little to no labeling in the placodal derivatives. And I've shown that with exemplar sections through some of the placodes along the anterior posterior axis. The FOXI3 lineage tracing begins at embryonic day 6.5. And when we look at later stage Cre activation, we see that the anterior placodes like lens and trigeminal ganglion do not show TDT labeling. Even the posterior placodal labeling, like the inner ear and this epibranchial ganglia, begins to diminish by embryonic day 9.5. And this correlates well with FOXI3 reporter expression, which, if you may recall, begins at embryonic day 7.0 and diminishes significantly by embryonic day 8.5 from the anterior head region. But what's noteworthy is that in any of these time points of activation, we still see robust placoidal labeling, some epidermal labeling, and little to none mesenchymal staining. And to rule out that there was no contribution to any of the other ectodermal lineages at any point of time in development, we verified TD tomato signal at an earlier stage of harvest at embryonic day 9.5, where we expect to see SOX9 positive streaming neural crest cells. And this section through the hindbrain, as well as the section through the forebrain, shows that the green SOX9 positive cells do not show TD tomato labeling, thus showing no contribution to neural crest or neural tube lineages. We also verified absence of TD tomato signal in the neural tube by identifying the region with TUB3 staining. So in conclusion for the data I've shown you so far, from the onset of expression, FOXI3 positive neural plate border only gives rise to placoidal and epidermal lineages. So what happens in absence of this gene? Our lab has reported that FOXI3 knockout mice lack inner, middle, and outer ear. The images from left to the right show absence of the otic vesicle labeled with PAX2, the middle ear ossicles identified with cartilage and bone staining, as well as absence of the pinna of the outer ear. These mutants also show lower jaw defects. However, for the purposes of my study, we focused only on the inner ear and other sensory placodes. So we ask, if the FOXI3 neural plate border contributes to all placodes and mainly placodes, what happens to the FOXI3 lineage in the null mutants? And for this knockout lineage tracing, we can use our FOXI3 CRE-ER line. The CRE-ER construct replaces the coding sequence, making it a null allele. So if we use our FOXI3 CRE-ER mice, breed in TD tomato homozygosity and fibmate them, we expect to find 25% of the embryos to be FOXI3 knockouts, double positive for CRE and no protein expression. We expect that in these knockout mice, FOXI3 promoter will still activate at the neural plate border, activate lineage tracing and the lineage tracer of TD tomato. However, no FOXI3 protein would be expressed. Thus, we can find what happens to the neural plate border cells in the null mutant. We can identify litter mates with genotyping for CRE-ER as well as phenotyping, where we will see absence of the otocyst and pharyngeal arches in the mutants, as highlighted here in this example. When we analyzed FOXI3 lineage in the knockout mutants, we find that FOXI3 lineage still is contributing to placoidal and epidermal derivatives. And you can see magenta TD tomato staining in the lens, as well as the olfactory placode in the mutants, as well as the controls. The anterior placodes like lens and olfactory pit are present and they're normal in morphology. However, the inner ear is absent and several cranial ganglia are absent and malformed as well. Furthermore, what is noteworthy is that we see increased lineage tracer or TD tomato labeling to the epidermis, as shown by arrows in these two panels. And we also see significant mesenchymal staining compared to the controls, as highlighted in asterisk. When we systematically look for derivatives of all four ectodermal lineages in our experiments, we see, as in the previous images I showed you, higher epidermal labeling in the knockouts compared to the controls, higher neural crest labeling, and you can see this in the section by the forebrain where the skull would form, and you can see double positive labeling of SOX9 positive cartilaginous progenitors around the forebrain that are also positive for TD tomato signal. And we also see patches of TD tomato labeling in the neural plate compared to absence of any such patches in the controls. So in conclusion, we see that the loss of FOXI3 gene expands the fate of the neural plate border, whereas in the control lineage tracing, the area only contributes to placodes and epidermis. However, in the mutants, it now contributes to all four ectodermal lineages. Or to rephrase, FOXI3 is required to restrict the fate of the neural plate border to placoidal lineages. So to summarize this study, FOXI3 labels the neural plate border. It is the earliest transcription factors specific for placoidal lineages identified thus far. This actually makes FOXI3 an excellent tool. And in the study, we have introduced 2 novel mouse lines to query, isolate, and precisely manipulate, let's say, signaling pathways or fate of the placoidal progenitors at the neural plate border. And in absence of FOXI3, ectodermal cell fate is not restricted and contributes to all 4 ectodermal lineages. So these are the key takeaways from the paper that our lab published in Development late last year. But in the next couple of minutes, I want to share some new unpublished data that I'm working on to develop as a part of my future independent research program. And for this, I go back to the question of cell fate specification at the neural plate border. Specifically, what is the transcriptomic variability within these cells? And I took advantage of a smart-seq-like sequencing technique called MacQseq, which is available at Baylor College of Medicine, to study mid-gastrulation embryonic and ectoderm. I isolated the anterior embryo and enriched my collected cell population for the lateral ectoderm, and I obtained around 10,000 unique transcripts and 150K reads per cell, providing me a very rich database to identify unique transcriptional signatures within the neural plate border. And I clustered these cells to identify those unique transcriptomic states within this patterning event. And using known transcription factors such as FOXI3, OTX2, TFAP2A, SOX9, and many more, I was able to label these clusters and identify and assign them potential cell identities. Of course, this is only transcriptional states, and cell fates remain to be determined for each of these clusters. However, I can shortlist likely gene candidates to study and not only add details to the known gene regulatory networks, but also perhaps isolate, manipulate specific ectodermal cell types, just like we did in our previous study with FOXI3. So I want to highlight 3 specific placoidal, 3 specific 4-kit transcription factors I find in my data set that are asymmetrically expressed: FOXD4, FOXC1, and FOXB1. And there is indeed reported evidence of each of these genes and their importance in formation of the anterior head tissue. And knockouts of many of these genes can lead to craniofacial defects. So more work needs to be done to understand roles of these transcription factors in neural plate border development. But with this data set, we can answer many, many questions. I can find transcriptional factors specific for each or a subset of the ectodermal lineages. I can study their necessity and sufficiency in the ectoderm, specifically for neural plate border cell specification. And I may also find competing transcription modules within an intermediate cell type to address the questions of cell competence and specification within the neural plate border. And with that, I want to give a shout out to my lab. We're a fun bunch here in Houston, Texas. And thanks to Andy, my postdoctoral mentor, Helen, Hongyan, and Hari. They were the co-authors that contributed to the FOXI3 paper, and Sue and Rachel, who were my mentees during my tenure here. And they both aided my projects, and all the other Groves Lab colleagues. Also, thank you to Yichi from Chuck Zhang's lab. They were the ones who did the MacQseq. They did the library prep, sequencing, and mapping for me, and several Baylor cores for their support. And thank you guys for your attention. And I'm happy to take questions. And you can also contact me on this email or Twitter.
Thank you, Ankita, for the wonderful talk. So we have around 10 minutes for the Q&A, and for the mark for the audience, please be reminded the Q&A can be written, and I will read it out for you. I have some question for you.
Yes.
I would say my background is not developmental biology, so I'm sorry if my question is a little bit naive, but the transcription factor that you are talking about, the FOXI3, do you have any kind of idea what is the downstream of those these transcription factor?
So studies from our lab has shown gene expression defects in knockout embryos in mice as well as chicken embryos. And we see genes like 6-1, ISAPSENT1, which are conventionally placoidal genes that are diminished in expression, as well as DLX5, also another placoidal gene, which are diminished in expression in the knockout.
Okay, we have a question from the audience. Very nice talk. Is it known which transcription factors are regulating FOXI3 in the neural plate borders?
I actually don't have an answer to that question, but we know that, so in context of inner ear, which is the primary focus of our lab, we know that FGF signaling from the hindbrain is important for patterning the ectoderm that invaginates to make the inner ear. And in our FOXI3 knockouts, the FGF signaling is not disrupted. However, FGF signaling defects do lead to inner ear defects. It is likely that FGF signaling is one of the potential upstream mechanisms before FOXI3.
Okay. If there are any more questions, thank Ankita for the wonderful talk, and I will turn the table to Katie for the second speaker.
Okay, thank you. All right, so yeah, I'm Katie Goodwin. I'm a postdoc in the McDowell Lab at MRC-LMB in Cambridge. And it's my pleasure today to introduce our next speaker, Dr. Konstantinos Kalyviotis from the Rosenblatt Lab at the Francis Crick Institute and King's College London in the UK also. So today he will be presenting work from his time in the Pantazis Lab at Imperial College London, where he is currently a visiting researcher and where he also completed his PhD in bioengineering by developing versatile tools for advanced optical precision imaging. So throughout his career, he has been an engaged member of the zebrafish and microscopy communities, as a presenter at conferences internationally, as an organizer of scientific meetings and workshops, as an elected member of the European Zebrafish Society Board, as a co-founding member and chairperson of the Young Researcher Committee of the European Zebrafish Society, and finally, as a recipient of several scholarships and awards, including the Christine Beattie Award from the International Zebrafish Society, and several awards for outstanding presentations at conferences. And so alongside carrying out his beautiful and cutting-edge research and alongside his extensive scientific community involvement, he has demonstrated a truly impressive commitment to teaching and mentorship, including supporting the work of nearly 50 junior researchers in the lab. And so with all that, I think we can agree that we're in for a real treat today. And I'll turn it over now to Konstantinos to teach us about his technological advancements and share his beautiful work. Please go ahead.
Thank you so much, Katie, for the wonderful introduction. And really, thank you so much. And today, first of all, before I start, I would like to thank the SDB and the organizers for giving me such a great opportunity to present my work. And really, thank you so much. And as Katie mentioned, I'm currently a member of the Rosenblatt Lab at the Francis Crick Institute and the King's College London, studying the biomechanics of cell extrusion. But today I'm going to present you my work from my previous lab, the Pantazis Lab at Imperial College London and the Department of Bioengineering. And more specifically, I'm going to present you our work on GENEPI, a highly specific and non-invasive reporter biosensor for visualizing piezo activity across biological scales. Measure what can be measured and make measurable what cannot be is a Galileo's phrase that could wonderfully summarize why we develop and we want to develop biosensors in biology. We want to make things measurable in order to be able to develop our models and understand how systems, our biological systems work. And as we are always interested in making invisible things visible, if I could say, we set out to develop a biosensor that would enable us to visualize how cells sense mechanical forces and mechanical stimuli. And of course, this led us to piezo1, but you would ask why piezo1? Of course, piezo1 doesn't really need an introduction after Wade's presentation 2 months ago and the Nobel Prize in 2021. But piezo1 is a mechanosensitive ion channel, which is expressed in a wide range of tissues. As you can see, it's almost everywhere, and is involved in a multitude of biological processes, from stem cell fate to cardiovascular homeostasis and red blood cell volume regulation and vascular and lymphatic development. So I would say it's a sensor for piezo1 would have a broad applicability. But you would say, how we can engineer such a sensor? Our design is based on the calcium microdomain hypothesis, which states that the calcium concentration near the vicinity of the pore of the channel is actually much higher when the channel is open compared to the closed channel. So naturally, we thought, okay, we can target this calcium microdomain of piezo1 with a genetically encoded calcium indicator, and then we can visualize this channel opening. And this is what we actually did. So we targeted the calcium microdomain of piezo1, and we were quite lucky at that point because the structure was not available, but the C-terminus of piezo1 actually resides near the vicinity of the pore. So we targeted the C-terminus of piezo1 with a green genetically encoded calcium indicator, a GCAM, and with two characteristics. First, low affinity, because we wanted to be sure that we are not going to sense intracellular calcium fluctuations, and the high dynamic range, just to be sure that we can distinguish the on-off states easily. Of course, we used also a linker, a flexible linker, and of specific length, just to be sure that we are providing specificity without affecting piezo1 structure. So we are still in the calcium microdomain, but we are we are not really affecting the structure of piezo1 and eventually its function. Sine, a wonderful PhD student in the Pandazis lab, performed a systematic screening for such a piezo1 specific biosensor. As you can see here, she tested 5 different GCAMs with different calcium affinities and dynamic ranges using different linkers. And she tested these 20 variants at the end for 2 different conditions. One for a piezo1 dependent stimuli, shear stress, so mechanical forces, and a piezo1 independent stimuli, ionomycin. We could say that in this setup, an ideal sensor for piezo1 specific readout should respond higher to shear stress compared to the cytosolic indicator, lower than the cytosolic indicator in the non-piezo1 piezo1 independent stimuli of ionomycin, and overall it should respond to shear stress more than it responds to the ionomycin treatment. Here you can see these screening results. And if you can see much easier probably in the summarized table, we had one variant, actually one piezo1 fusion, which have piezo1, one linker repetition, and then GCAM G4 that actually fulfilled both our key requirements. And we named this variant as GENEPI, as genetically encoded piezo1 indicator. Actually, the story is a bit more funny because Ali, another former PhD student in the Pandazis lab, got inspired by the herbal liqueur GENEPI to name actually a GENEPI. And you can see actually that this was one of the presents, gifts that I got from my supervisor, Lucky, here for passing my Viva. The first thing that we wanted to know, because we are actually targeting a functional ion channel, was to be sure that we are not really affecting its functionality. So we performed a series of electrophysiology experiments. And as you can see here, GENEPI has similar pressure-dependent responses to the wild-type piezo1, and also retains the typical conductance of piezo1 channels at around 25 picosiemens, as you can see here. And of course, it responds to the piezo1 specific agonist which is YODA1, as you can see here. But it does not only respond, but it seems to respond actually in a dose-dependent manner, as you can see here, which doesn't mean that actually more calcium, let's say, is there, but actually what we believe our current model right now is that having higher concentrations of YODA1 actually activates more channels in the membrane, more piezo1 in the membrane, which then leads to the increase in the fluorescence intensity. But someone would ask, are you sure that this increase in the fluorescence that you see is from calcium flowing through the channel? To answer this question, we used a similar setup that Arden Pataputian used when they were screening for YODA1, which is very simple. So we actually chelated for the extracellular calcium. And as you can see here, in the presence of EGTA, even when there is YODA1, although in the normal conditions, GENIP responds to YODA1, when there is EGTA, there's no response of GENIP, which actually confirms that the GENIP response is due to extracellular calcium flowing through the channel, showing the piezo1 activity. Of course, up to this point, someone could say, okay, you're saying that it's flowing through piezo1, but are you sure that this is piezo1 specific and not, for example, because in the membrane there are different other ion channels, are you sure that they are reporting only the piezo1 signal? To answer this question, we focused on the P2X channels, which they have very similar conductance to piezo1 at around 20 picoseconds, if I'm not wrong. And as you can see here, when we stimulate the P2X channels with ATP, we see the increase in the cytosolic calcium indicator here, when there's ATP, while GENIP at the same time does not respond, which actually confirms that the GENIP response is not affected by other nearby calcium channels, and more specifically, I would say, from their calcium microdomains. So after this initial, let's say, in-depth characterization of the biosensor GENIP, we wanted to use GENIP in order to visualize piezo1 dependent activity in different, let's say, setups, going from the subcellular level to the tissue level, and of course, at the end to the organismal level. And first, we focused on the subcellular level. So, and I will show you here from the autonomous cellular forces. We performed time-lapse total internal reflection fluorescent microscopy in cells expressing GENIP, which actually visualizes, let's say, around 200 nanometers of the membrane, which is attached to the glass. And as you can see, and I hope that you can appreciate in this short video, we see that these piezo clusters, these GENIP clusters, they're quite dynamic. So we tracked more than 5,000 piezo GENIP clusters, and then we performed the mean square displacement analysis. And we saw that GENIP mainly diffuses in the membrane of the cells, as it had been shown for piezo1, and has a diffusion coefficient of around 0.005 micrometers squared per second, which is again in the range of previous reported values for piezo1. But someone would say, okay, now you have GENIP, you seem to track it, which is something that you could just do with GFP tagging. What is the extra thing that you can do with GENIP? So the extra layer of information that we can actually get out of our systems now is that we can have a functional readout. And you can see here, for example, that individual classes, we can actually now extract their dynamic activity of these clusters. And as you can see here, this cluster shows a dynamic activation here, while the other classes doesn't seem to respond at the same time. Of course, this means that using the right segmentation algorithms, you can actually have a parallel analysis of individual GENIP clusters in a whole cell. And here I'm just showing you 49 of them in the same cell before. So after doing this analysis of the GENIP dynamic activities in the subcellular level, then we moved to the tissue level, and we focused on the autonomous beating of cardiomyocyte microtissues. So we were hoping that would be something, let's say, contraction triggered. To this end, we generated a mouse embryonic stem cell line in which GENIP is under the control of a TETON promoter. So after we confirmed that we can induce GENIP in these mouse embryonic stem cells by adding DOX, and also we confirmed that GENIP is functional within them by adding YODA, Then we differentiated these mouse embryonic stem cells into 3D cardiac microtissues, which I hope that you can see here that they were contracting nicely. And of course, these microtissues have multiple beating cardiomyocytes within them. So we express GNP in these cardiac microtissues, and we will wonder whether we can see actually some contraction-triggered PSO1 activity. And this is what we saw. We saw actually two types of activities. When we were just imaging whole microtissues, you would see, let's say, systemic GNP responses, which would coincide with the contraction of the nearby cardiomyocytes, as you can see here. But when we dissected individual, let's say, patches of beating cardiomyocytes, we saw that we have some local responses. And here you can see, for example, in the asterisk, we have a cardiomyocyte which is beating. And I hope that you can appreciate that this side of the GNP expressing cell is actually responding on each contraction, while the other side, which is from the other part of the cell, does not really respond. When we looked more into this type of responses, we actually saw that these local responses, and of course also the systemic responses in general, they are triggered from the local membrane deformation due to the contraction which is applied to the membrane of the GNP expressing attached cell. Of course, this means that because we see a bit of variability in the PSO1 activities that we get, that higher forces would lead to greater membrane deformation, which of course then would lead to higher fluorescence intensity. So after doing these experiments and seeing how GNP works in the microtissue environment in the tissue level, then we eventually wanted to move to an organismal level and see PSO1 dependent activity in vivo. To this end, we generated a GNP transgenic zebrafish in which GNP is under the control of a Hitchcock promoter, which would give us two things. First, spatiotemporal control of expression and homogeneous expression in the whole zebrafish, which was a key at that point when we were actually testing GNP. The Hitchcock protocol is actually very simple. You can just put your embryos into a falcon tube. We usually grow them at 29 degrees. So we can Hitchcock them at 37.5 degrees for around 1 hour. And usually we put them back at 29 degrees, and around 4 to 5 hours later, we can screen for positive embryos. And I just want also to mention here that our data up to this point show that these transgenic GNP fish are actually healthy. They seem to reproduce normally as well, which is a key. Upon Hitchcock, it seems that we reach a steady fluorescent state at around 4 to 5 hours post-Hitchcock. And as you can see here, we get a homogeneous expression across the whole zebrafish embryo, which is actually quite around 2 to 3-fold increase in the total fluorescence in the embryos. At that point, it was actually very encouraging when we were imaging different tissues, that GNP was mainly localized in the membrane of the cells, which is what someone you would expect for PSO1. Okay, we express GNP now in the fish, but are we sure that's actually functional? To test this, we performed a series of experiments in which we transferred fish from the normal medium that we grow the embryos into solutions that they are containing Yoda-1, which is the PSO1 agonist, in the presence or absence of ruthenium red, which is a generic ion channel inhibitor. And then we were washing them out to E3 medium. As you can see here, now the GNP expressing fish, when they are transferred to Yoda-1, there's an increase in their fluorescence, while when we transfer them to a solution containing Yoda-1 and ruthenium red, their fluorescence was going down. So when we going to ruthenium red, it was going further down. And really impressive, it was that when we were washing them out, we were just going to the pre-treatment levels. And this was something very impressive because it was really happening instantaneously the moment that we were washing these embryos back to their E3 medium. After confirming now the functionality, we were really wondering whether we can see PSO1 dependent activity under physiological conditions in vivo. And to this end, we focused to the zebrafish beating heart. And we performed high content imaging, and then we reconstructed individual bits. And you can see here a video that was produced after doing this processing. And I hope that you can appreciate that there is quite dynamic, let's say, activity of piezo1 in the zebrafish beating heart, and especially in the center, in the region of the anteroventricular canal, where the valve will develop later on. After confirming that GNP is expressed in both tissue layers of the zebrafish beating heart, so both the endocardium and the myocardium, we then focus on trying to analyze the activity profiles of GNP in the atrioventricular canal when it seems to have, let's say, more interesting activities. And we found that it has this biphasic response in individual cycles, which was actually abolished when we stopped the heart with BDM. So at first, this was actually quite curious because we were expecting to have something like what we have in the cardiac microtissues experiment, so single peaks because of the contraction triggering the activity of GNP. But as we were actually, just analyzing further the data and taking a more close look, we understood that actually this pattern, this biphasic pattern, is very specific. The first peak seems to coincide with the atrium contraction, and the second peak seemed to coincide with the ventricular contraction. And if you consider that at this time point, there's no valve, so there's bidirectional flow of the blood, we wondered whether this is actually something, the GNP responses that we see, whether these are due to shear stress, so mechanical forces. So what we found in the literature is actually that the wall shear stress measurements at the same time point in the zebrafish heart actually have this same biphasic, let's say, pattern, which is very similar to what we get with the GNP response, which up to this point, our current hypothesis is that this GNP response is actually due to the shear stress forces from the blood flowing through the heart, the zebrafish heart. Of course, this new line, this zebrafish line, opens up a lot of possibilities and a lot of opportunities for questions. And I'm here, I just put here some spontaneous piezo1 activity, which we see in the developing zebrafish forebrain. So you can see that some of the cells, they show some local activities on the side, or some whole cell activities, which are actually statistically significant between the different, let's say, cells. And they kind of look like to be like salt and pepper responses. Of course, their role still need to be investigated, but now that we can actually see piezo1 activity in vivo, this would give us a lot of opportunities to answer a lot of questions about the role of piezo1. To sum up, GNP is a fluorescent biosensor for visualizing piezo1 dynamics from the single cell level to that of the entire organism. And we hope, and I hope personally, that you will use, and we are all going to use GNP in order to study the piezo1 dependent activity and the role of piezo1 in developmental disease. Of course, currently there are a lot of other, let's say, engineering to be done. Let's say, for example, to have some other color variants that will enable us to do multicolor imaging. And currently, we have also developed in the Pantasis lab transgenic mice with endogenous tagging of piezo1, which kind of is like the holy grail, let's say, to see the dynamics of piezo1 happening in live in physiological expression conditions. And we have also in collaborations that they develop transgenic flies expressing GNP. And we hope that all these lines and will actually democratize the study of mechanosensation, making it more accessible to all the labs using such biosensors. Before I end my presentation, I want to briefly say and point out that the GNP design of targeting the calcium microdomain of an ion channel could be actually a blueprint for engineering other ion channel biosensors without affecting their function. Although we believe that the key thing is actually to have a low affinity ion indicator of high dynamic range, and of course, using the right linker. Now, at the end, I would like to thank Lucky Pantazis, who was my PhD supervisor for, and later on worked in his lab for my first postdoc, for supporting all my crazy ideas. And of course, all the Pantazis lab members, and of course, Sine, who started the work on GENEPI. We should not forget the zebrafish, who gave us such beautiful results. And of course, our great collaborators, not only from the UK, but also from around the globe. Finally, Department of Bioengineering at Imperial College London, which funded my PhD, and the Royal Society, which funded my first postdoc in Lucky's lab. And with that, also, I would like to thank you for hearing my presentation. Thank you so much.
Thank you so much, Konstantinos. So our audience members, you please feel free to add questions to the Q&A. But I'm trying to decide which question I want to start with because I have several. One of the things is sort of like you're able to track piezos at a subcellular scale, and it's not quite a reporter. You're not just tagging it as a sensor, as you said. So the fact that you can see so many of them suggests that there's sort of a baseline level of openness across the population. Do you see little spots disappear completely and come back? And part of that question is also, do you ever see the intracellular piezo that was published by the Rosenblatt lab in 2017, where they see all that piezo inside the cells? Because I've never seen that. So there you go.
That's really great questions. So I can tell you something that's very interesting because of the baseline, let's say, fluorescence. So something that we notice in general in our turf experiments and also in the zebrafish that I showed you is actually that you see a lot of green happening, right? So we believe that there is kind of like a very basic piezo1 activity baseline, which it's almost all the time happening because of the mechanics of the cells. And it was very interesting when we were doing the BDM experiments, actually, that at some point they were just making the cells really loose and the cytoskeleton is actually really disrupted, that this baseline signal was actually going down, which means that there is kind of like a baseline piezo activity being kind of, let's say, open on the, not all the time, but in the cells. Now, regarding the tracking, I can tell you that because, the GCAMs, they're not really perfect. There is a bit of background fluorescence, but of course, dynamic range means that their own state is actually much more easy to see than their off state. So we kind of get, let's say, you can even track the cluster, which are quite big. They're not really individual piezo clusters. I need to mention that probably there are hundreds of them there. So you can actually track the clusters from the background fluorescence.
Great, awesome. Okay, so we do have a question in the Q&A. So what is the reason behind making the GenEpi fish a heat shock?
So can you say again? Sorry, I cannot hear.
What is the reason? I think it's a question about heat shock aspects of the zebrafish line.
Yes. So
yeah.
Yes. So we first, because of that point, you have to consider that our goal was to mainly characterize GenEpi. So try to understand how we can actually get meaningful readouts from this biosensor. So we knew that this is actually functional. It gives piezo specific readouts, but we wanted to have, let's say, something like really homogeneous expression, which is something very difficult to manage to have in zebrafish. So, for example, even UB or beta-actin promoters, you still have kind of like a patch expression, so not really same expression levels in different cell types, which we wanted to kind of like overread. So that's why we selected the heat shock promoter, because we want to have, let's say, temporal control of the expression, really ramping the expression high in order to be able to visualize it, and also have, let's say, homogeneous expression that enabled us also to do all the experiments for the in vivo functionality of GenEpi.
Okay, great. We have another zebrafish question. So I see you used long fin zebrafish for the heat shock experiment. Is there a reason you chose that line of fish?
Not really. This was the wild type line that we had at that point. So we did the injections. This was actually the zebrafish. We initially started the work with the Scott Hawley lab in Yale, and with Dorte, and the wild type line that a lot of people used was the long fin. So we performed the initial experiments in long fins.
Okay, we have another question here. So what is the result of loss of piezo-1 in zebrafish? Are they embryonic lethal?
So that's a very good question, because actually the loss of piezo-1, there's a really nice work from the Diestanes lab that they studied the role of piezo-1 and piezo-2A in the heart development, and last year I think, or 2 years ago. But the piezo doesn't seem to be lethal, the knockout. But when we do knockout in the mice, we get they are embryonic lethal, so they are dying actually when the vascular is starting to developing, not really early in development, which means that probably it's not that interesting for us. We were hoping that would be really much earlier, but it's not.
Okay. I don't see any other questions in the Q&A, so I'm going to ask another question. In sort of deploying these biosensors in tissues and in embryos, especially if we start moving into less optically favorable embryos like mouse, do you anticipate any issues with imaging deep into samples with this fluorescence-based sensor?
Yes, that's our main problem right now, because we are now trying to evaluate the transgenic mice that we have, which has the endogenous piezo-1 tagged with the GCAM that we used, the same approach. And what we are trying to do right now, and we are hoping to do, is to make ex vivo tissue, ex vivo live tissue imaging, because it's quite difficult to reach really deep and image well. And we have to consider also, depending on the expression levels of piezo-1, this is it could be even more tricky in different cell types.
Okay. Okay, cool. So there are no other questions. So at this point, I'd just like to thank both of our speakers today, Ankita and Konstantinos, for their excellent talks. This seminar has been recorded and it will be available on the SDB website next week. And this is the last Ethel Browne Harvey Postdoctoral seminar of the season. So I'm now going to turn it over to Martha, the SDB postdoc representative. Please take it away.
Thank you, Katie. So we want to thank all the postdocs that presented and moderated this past year. All of their talks are available on the SDB website. And I want to remind you that the next season of the Ethel Browne Harvey Postdoctoral Seminar will kick off on September 13th, and the applications will open in June. So thank you, all of you, for coming.